Okay, so we talked about trophic groups and we talked about how they got carbon and how they made their energy. Now I want to look just a little bit closer into energy. So we talked about ATP cycling between catabolic and anabolic reactions, exergonic and endergonic reactions, and how ADP is phosphorylated into ATP. Now I want to talk about how ATP is made. ATP is made in the electron transport chain, and I really like the way this particular video demonstrates how ATP is made. So you're going to watch a video within a video. So when we talk about catabolic and anabolic reactions, and we talk about how catabolic reactions release energy, and that energy is used to form ATP, it's not like in the cell ADP is actually sitting right there next to a catabolic reaction with its inorganic phosphate ready to be pushed on to make ATP. It actually has to happen in the way that we just showed with the video. So we have to actually make NADH and FADH2 so that we can create that proton motive force across the cell membrane in order for the protons to come back through and drive ATP synthase. And so next follows, where do the cells make NADH and FADH2? Those are made when we actually break down chemical bonds in catabolic reactions. So we're going to end up at this electron transport chain, but I'm going to circle back to this because we're going to start at the beginning now. This is actually the end, and we're going to start at the beginning now. So remember, exergonic reactions release potential energy. So exergonic reactions release energy, which allows ATP or ADP to be phosphorylated, but that's going to happen through the electron transport chain. But first we have to go through glycolysis, the Krebs cycle, and FADH and NAD have to be reduced to FADH2 and NADH, and then those go to the electron transport chain. So we're actually in this process, we're talking about respiration. And respiration is not breathing. Respiration is when we are making ATP using an electron transport chain. There is aerobic respiration, that's what we do, and the video was of aerobic respiration because the final electron acceptor at the end of that electron transport chain is oxygen, and so there's gas exchange there. But we're actually going to talk about both aerobic and anaerobic respiration. Both of those end with an electron transport chain. So let's start at the beginning. Let's start with glycolysis. Glycolysis is glycolysis. So we're going to take glucose, which is that 6-carbon molecule, and in glycolysis we're going to break it down into 2 molecules that each have 3 carbons, and those are called pyruvate. In the process of glycolysis, we're going to release 2 ATP. So actually, this would be a really good time for you to pause this video and print out that handout that's on Blackboard and start filling that out. Because our very first pathway on that handout is glycolysis, and our very first molecule is glucose, which is a 6-carbon molecule, and the product of glycolysis is pyruvate or pyruvic acid, and that is a 3-carbon molecule. We also get 2 ATP out of glycolysis, and all of those intermediate steps and things like that, I'm not going to ask you to memorize. I had to memorize those in graduate or undergraduate school when I was sitting in your seat, and I'll tell you right now, I couldn't remember a single bit of it. So it doesn't make sense to ask you to memorize it also. But get that sheet out, fill that in, because that's going to help you kind of keep track of all of these things. Okay, so starting molecule is glucose, first pathway is glycolysis, first product of that pathway is 2 pyruvate, which each have 3 carbons, and 2 ATP. Now we're not done. We still, we have 1 NADH. That can go to the electron transport chain. But if you remember in the electron transport chain video, that hydrogen atom gets broken into 1 proton and 1 electron. 1 proton hardly makes a proton motive force. 1 proton isn't enough, right? That's going to make ATP synthase turn just 1 time. So we need to make some more NADH and FADH2. And so what we're going to do next, and we're still just focusing on aerobic respiration at this point, is we're going to go to the Krebs cycle. So we're going to go from pyruvate, which is a 3-carbon molecule, and we're going to run that into the Krebs cycle. Now the Krebs cycle is going to start with something called acetyl-CoA. Acetyl-CoA is a 2-carbon molecule, and so we're going to lose a little carbon dioxide along the way. This is our first place where carbon dioxide is released. So we've actually popped off a single carbon from glucose. We've broken that carbon bond. This is why we exhale carbon dioxide. But the real reason why we exhale carbon dioxide, and the real place where we make the most NADH and FADH2, is in what's called the Krebs cycle. So we had this little intermediate step where we converted pyruvate into acetyl-CoA. And acetyl-CoA is a 2-carbon molecule. Now, the Krebs cycle is called the Krebs cycle because actually the first thing that happens in the Krebs cycle is that acetyl-CoA gets smashed with the product of the last turn of the cycle. And the product of the last turn of the cycle is oxaloacetate, which is a 4-carbon molecule. So when you have a 2-carbon molecule and a 4-carbon molecule that get smashed together, you've created a 6-carbon molecule, citrate, on the right there. And that 6-carbon molecule now has several bonds that we can break apart. We can pluck off those carbons one at a time. We can rearrange those molecules, and each time we do that, we are releasing a little bit of energy. So if we go along the way, we have isocitrate and alpha-ketoglutarate, and if you notice, we've plucked off a carbon molecule. Alpha-ketoglutarate only has 5 carbons, and when we plucked off that carbon as carbon dioxide in step 3 there, we've created an NADH. And then we pluck off one more carbon and we create one more carbon dioxide, and we've created one more NADH. And then we can actually rearrange the molecule a few times, and we can make FADH2 by rearranging it, and we can rearrange it one more time, get another NADH, and now we've got oxaloacetate. This is why we exhale carbon dioxide, and this is where we get the most NADH and FADH2 from glucose. I had my cell biology professor who actually made me memorize all these things. He always said, and this is how you need to lose weight if you want to lose weight. We take in carbon, and the only way for that carbon to leave your body to have less carbon mass is for you to exhale carbon. Right? So it really does make sense. You got to work out if you want to lose weight, because that's how we lose carbon, is by exhaling it as carbon dioxide. Okay, so takeaway from this, you got your sheet out. Okay, starting molecule or second pathway is Krebs cycle. Starting molecule of the second pathway is acetyl-CoA. It has 2 carbons. Our end product, our most important end product, why we did all this, was so that we could get lots of NADH and FADH2. We also get carbon dioxide, which I've emphasized a little bit here, and we also get oxaloacetate, because that molecule is what's going to get smashed with the next incoming acetyl-CoA. Now we've got several NADH and FADH2 molecules that can go down to the electron transport chain and donate those hydrogen atoms to be split into protons to create a proton motive force. So in eukaryotes, this is all going to be happening in the mitochondria. The mitochondria has all kinds of inner foldings of the inner membrane. It's called cristae in mitochondria, so that we can have lots of electron transport chain enzymes all lined up, and we have all kinds of periplasmic space, or I'm not even sure what it's called, in intermembrane space in mitochondria, so that we can have those protons all out there driving that proton motive force. So the NADH and FADH2 drop off the hydrogen. The hydrogen gets split into a proton, which is H+, and an electron. The electron gets shuttled from molecule to, or enzyme to enzyme. These are called cytochromes. And every time one of these cytochromes, or a few of them, get an electron, they use that to grab another proton and throw it across the membrane to create that proton motive force. So what's most important about the electron transport chain isn't really the electrons, it's the protons. The proton motive force, all of those H+s, they want to go back down against their gradient, or down and equalize their number, so they want to pass through that ATP synthase. And so on the bottom of the screen there, ATP synthase is kind of upside down. They're going to go from the bottom of your screen up through ATP synthase back into the matrix, the inside of the cristae. When they go back through, that physical kinetic movement is what allows that enzyme ATP synthase to smash that phosphate onto ADP. So each time one proton goes through, that thing turns one time, and it makes one ATP from ADP. Now, here's the fun part. those electrons have to go somewhere at the end. Otherwise, they're going to back up the whole assembly line, and you're not going to get ATP. And so you need to have a final electron acceptor. And our final electron acceptor is oxygen. It's not the oxygen we breathe, because the oxygen we breathe is O2. It's 1/2 oxygen, so it's one single oxygen atom. And, fun fact, those protons get reunited with their electrons. They become hydrogen again. And so 2 hydrogen plus 1 oxygen makes water. That's why when we exhale, there's so much moisture in our breath, because we are also exhaling water. So we exhale carbon dioxide from the Krebs cycle, and we exhale water from the electron transport chain. Isn't the body just amazing? Now, where does all of this happen in a bacterium? They don't have mitochondria, right? So this has to happen in the electron transport chain. The electron transport chain is probably the most important thing that the cell membrane does for a bacterium. And just for funsies, is this bacterium gram-positive or gram-negative? This one's gram-positive. So notice, a gram-positive bacterium still has to have periplasmic space. It's just very narrow compared to a gram-negative. Okay, so we talked about aerobic respiration. Now let's talk about a couple of the little things that we go along with aerobic respiration. Remember those enzymes in the electron transport chain? Those are called cytochromes. And in fact, one of those cytochromes, the little teeny pink one there that's kind of high on the picture, the little teeny pink one next to the red one, between the green one and the red one, that's called cytochrome C. And some organisms, some bacteria, have a different type of electron transport chain. So they don't have cytochrome C, which means that they don't necessarily use oxygen. They don't require oxygen. We have cytochrome C because we require oxygen. Some of them can have something else. And so part of identifying bacteria is looking to see if they require oxygen for respiration. And we're actually going to do this test. This is called the cytochrome C oxidase test. So the oxidase test tells us if an organism has cytochrome C. And if it does, then we know it requires oxygen for respiration. If it doesn't have cytochrome C, it might still be able to use oxygen. It just doesn't require it. It uses an alternate pathway. Another fun fact, cyanide kills by blocking cytochrome C. So if you were to take cyanide, you would actually suffocate on a molecular level because you would not be able to use oxygen as your final electron acceptor. Okay, so let me ask this in a different way. If an organism were to test negative for the oxidase test, what can you conclude? You can conclude that it does not require oxygen. It is not an obligate aerobe. It might use a different pathway. Other important things we need to think about when we're talking about identifying bacteria based on their respiration requirements. Oxygen can actually form different molecules in the cell or in the environment. If you require oxygen or if you live in an environment that has oxygen. And these are super damaging molecules. Things like superoxide, which is oxygen gas with an extra electron, and peroxide, such as hydrogen peroxide. Hydrogen peroxide is the most simple peroxide, where you have an oxygen-oxygen double bond. We can actually test to see if organisms produce enzymes that can help them deal with these very damaging molecules. So, for example, there is an enzyme called superoxide dismutase that will actually break down superoxide so that it is no longer so very electronegative. It doesn't have that extra electron. It's not quite as damaging. This guy is so electronegative, it'll actually pull electrons off of other molecules. Oh, I'm sorry, it will actually deposit electrons. Oh dear, now I'm making no sense. Reset. There's also a catalase enzyme. So we have superoxide dismutase, we also have catalase. Catalase is going to help organisms deal with peroxides, for example, hydrogen peroxide. So where am I going with this? I do have a point. The catalase reaction is a really simple first step reaction to help us differentiate between Staphylococci and Streptococci species. So for example, this is a strep throat plate. So I would make plates for the MedTech program, and I would take swabs from my throat, and I would spike in pathogens. And so I would spike in Streptococcus pyogenes, which causes strep throat. The problem with this is, as we'll learn in lab, some of us, including me, are natural Staphylococcus carriers. And so I didn't know when I was making these plates and I was culturing my resident flora that I also had Staphylococcus aureus. And both Streptococcus pyogenes and Staphylococcus aureus can break down red blood cells. Pyogenes does it, and it's very virulent. That's why strep throat is so painful. Staph aureus, when you're a carrier, you don't know it, you don't feel it, and I didn't either. So unfortunately, when I gave these plates to the students, and they had these organisms that could break down red blood cells, they had to be able to tell the difference between them. Now you could do a gram stain, and I've got gram stain pictures here. They're both gram-positive cocci, and as you know from lab, the arrangement can help you. You could have a Staphylo arrangement or a Strepto arrangement, but they're not always this perfect, right? Sometimes it's harder to tell. And so what you can do is you can take a little toothpick and you can smear the organism onto a slide, and you can take peroxide. Let me go back a slide. You can take regular old hydrogen peroxide from the grocery store, and you can drop it on that slide, and if the organism has the catalase protective enzyme, it will fizz. It will actually break down peroxide into water and oxygen. Hydrogen peroxide, H2O2, into water and oxygen. And so it'll fizz, it'll bubble. That's why when we get a cut and we put peroxide on it, it fizzes and bubbles, because we have organisms that have that protective enzyme, and our blood also has that protective enzyme. So we're looking at these two organisms, catalase positive and catalase negative. You got Streptococcus from strep throat, and you got Staphylococcus, which is usually on our skin, right? Staphylococcus. Which one do you think is going to be the one that has the catalase protective enzyme? The one from your skin or the one from your throat? The one from your throat where there's lots of carbon dioxide, or the one from your skin where it's bathed in oxygen all the time? Yeah. Staphylococci species are the ones that have the catalase. Streptococci do not. So it's a real quick and cheap, easy way of telling the difference. Okay, so we talked about aerobic respiration. We can tick that off our list. Now we have to think, well, what if there is no oxygen? Can we still keep running glycolysis? Can we still keep running the Krebs cycle? Can we still keep running the electron transport chain? Things are going to start backing up, right? We're going to have a problem. So organisms might use something different. They might use a different inorganic electron acceptor in their electron transport chain. So let me go back. Anaerobic respiration is still respiration, okay? So you are still going to have glucose as your starting molecule. Your first pathway is still going to be glycolysis. You're still going to get pyruvate. You're still going to have that intermediate step to make acetyl-CoA. You're still going to have a second pathway of the Krebs cycle. You're still going to make all kinds of NADH, FADH2, carbon dioxide, and that oxaloacetate that gets smashed back to the incoming acetyl-CoA. You're still going to run an electron transport chain, but if you notice, these organisms are going to use something other than oxygen. Now, oxygen's inorganic, and these example molecules, sulfate, nitrate, carbonate, are also inorganic. So, anaerobic respiration is going to use a different inorganic terminal electron acceptor other than oxygen. And so why is that important? Well, that's important for things like bioremediation, crop yield, and global warming. So nitrate, for example, if an organism can use nitrate as its final electron acceptor, it can actually contribute to the loss of nitrogen from soil, pushing that nitrogen into nitrogen gas, which goes into the air, which is a problem then, because if you have less nitrogen in the ground, you're going to have less crop yield and productivity. Remember, all organisms need carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. If they were to use sulfate, for example, these Archaea species, Desulfibrio, we can actually use them to our advantage. So, for example, there's a chemical company that is making this really acidic, toxic sulfate as part of its waste product, and it can't release that into the air. You can't just irresponsibly release this awful gas into the air. And so what they can do is they can actually run that gas through a anaerobic chamber. So all of these blue foam pieces there, those cubes, are going to have microbes growing on them. We've created a lot of surface area for biofilms to form, and they've sealed it up so that there's no oxygen, and these organisms are going to use sulfate as their final electron acceptor. And then I've got the chemical reactions there, but the point of it all is the sulfate actually gets converted into something that is now sodium bicarbonate, which is just baking soda, and that is absolutely safe to release into the air or the ground, and it's no longer toxic, all because those organisms are changing it via respiration. And then my last example would be our methanogens again. So remember, if they use carbonate or carbon dioxide, they're converting that into methane. They're generating methane. Methane is actually 19 times more powerful than carbon dioxide in terms of global warming potential. 19 times more powerful. So it's not just cow farts that are creating global warming, and it is not just, although it is almost certainly majority human influence, but we also have to think about our methanogens, our archaea that are converting these carbon molecules into methane, because they are also contributing to the global warming potential. And so those are just the 3 examples that are in the book, but the truth is that microbes have a lot of options. They can use things like iron even, or other inorganic molecules as their final electron acceptor. So they can be aerobic respiration, anaerobic respiration, and then we'll also talk about some of the other things they can do. So in aerobic respiration, we get 38 ATP, and that is the highest payout of ATP. In anaerobic respiration, you get somewhere between 2 and 36, and the range is because we don't know what final electron acceptor they're using. Depending on what final electron acceptor they're using, that'll say how much ATP, just how many times that ATP synthase can turn. But if you would take a note here, aerobic respiration is the biggest payout. Even the highest payout of anaerobic respiration is still lower than aerobic. You still only get at most 36 ATP. Okay, so now we can say that our respiration is finished. We've talked about aerobic and we've talked about anaerobic. Now, if Escherichia coli is usually using glucose in a phenol red basic broth tube covered in oil, so there's no oxygen diffusing in, that utilization of glucose is done through the process of, it's actually going to be done through fermentation. So we need to talk about fermentation. Now we did our yogurt lab, so we did a little bit about fermentation, but now we need to look at it a little bit closer. Fermentation is different than respiration in a lot of ways. Okay? So we don't have this exchange of gas. And remember, in respiration, we're using an inorganic electron acceptor, either oxygen or some other inorganic electron acceptor. In fermentation, we use an organic molecule. And actually what we use in fermentation is pyruvate. That's our final electron acceptor. So let's really examine fermentation here. You got your sheet out, right? We're going to start with glucose. We're still going to run through glycolysis. We're still going to make those 2 ATP, but now we're not having an exchange of gas, so we don't have an electron transport chain. So there is no reason to run the Krebs cycle and make NADH and FADH2. We're going to count only on those 2 ATP that are made in glycolysis. And the only way that we can make more of those ATP is if we can get rid of pyruvic acid or pyruvate. We have to be able to get rid of pyruvate in order to keep running glycolysis over and over and over to get 2 ATP. And the way you can get rid of pyruvate is by using pyruvate as the final electron acceptor. So remember that NADH that we made in glycolysis. We can actually take that electron, right, the H gets converted into a proton and an electron, and we can take that electron from NADH and we can actually dump it directly onto pyruvate, and that gets converted into lactic acid. So pyruvate's organic, and it is the final electron acceptor in acid fermentation. Or you can convert pyruvate into acetaldehyde, which is a 2-carbon molecule, and you can dump an electron onto that, and you can create ethanol. So in fermentation, there are 2 options. There is an acid pathway, and there is an alcohol pathway. Excuse me. Notice in the alcohol pathway, you get 2 carbon dioxide or a carbon dioxide coming off of that when you convert that pyruvate into acetaldehyde and then dump the electron onto it. So that is why when we have beer, it's carbonated, or champagne, it's carbonated because we're running alcohol fermentation. Okay, so as you know, fermentation is important in food and beverage industry. It is also important in chemical and pharmaceuticals. So there's an acid pathway and there is an alcohol pathway. And I know that last figure showed lactic acid, and certainly you get lactic acid, but you get other acids too. So if we look on the far left, if we dump an electron onto pyruvate, we can get propionic acid. Propionic acid is what makes Swiss cheese that really kind of funky Swiss cheesy flavor. Carbon dioxide is actually made in that particular pathway, and that's why you get those big bubbles in Swiss cheese, those holes. Certainly you know lactic acid because we made yogurt. So it's also cheddar cheese, soy sauce. If we move over to Saccharomyces, like Saccharomyces cerevisiae, which makes beer. That's also our bread and our wine yeast. So that's our yeast representative. All the rest of these are bacteria, but there's lots of organisms that can ferment. These are just some examples. Look at Clostridium. You know Clostridium, right? Botulinum difficile, perfringens, tetani. These guys can also ferment, and when they ferment, they make things like acetone and isopropanol, which is rubbing alcohol. They also make butyric acid, which is what causes gangrene. That cell death is from butyric acid. And then Escherichia coli and Acetobacter, they make acetic acid, which is what makes vinegar. So remember, Louis Pasteur was very interested in what makes wine or beer versus vinegar. Well, it's because it's a completely different organism. In fact, it's a completely different domain. Saccharomyces is yeast. Escherichia and Acetobacter are bacteria. So we actually have lots of options for fermentation, and these are even just a few. So fermentation, if you got your sheet out, still start with glucose, still go through glycolysis, but now those two ATP and that NADH get pretty darn important, because now we're going to go through fermentation. And you're going to have an acid pathway or you're going to have an alcohol pathway. And the starting molecule is going to be pyruvate for the acid pathway, and it's going to be acetaldehyde for the alcohol pathway. And your product is going to be an acid or an alcohol. And your final electron acceptor is going to be pyruvate or acetaldehyde. It's an organic molecule. It's pyruvate or acetaldehyde. And you only get 2 ATP. So now we'll call fermentation done too. Okay, I know this video is getting long, but we're going to wrap up with amphibolism. Amphibolism, amphi, right? Both directions, right? We've been talking about catabolic processes so far, right? And remember, metabolism is the sum total of all metabolic reactions within a cell. So it's catabolic and anabolic. And with our catabolic reactions, we actually use glucose as our starting molecule, but we can actually use other energy sources. Right? So we can use other carbohydrates, we can even use proteins and fats, and we can run them through those catabolic processes to still make ATP. And that's what amphibolism is. And so these terms are terms that we use with amphibolism. It's meaning going in multiple directions. So for example, gluconeogenesis means taking pyruvate and making glucose from pyruvate. So maybe we need to make a polysaccharide within the cell, and we've got pyruvate. We can actually turn pyruvate back into glucose and make a polysaccharide like peptidoglycan. I don't think we would. Or a glycocalyx of some sort, but something like that. Beta oxidation is when we take fats and we run them into the Krebs cycle directly. So acetyl-CoA is actually part of a fatty acid. And so if you can take that and you can take that acetyl-CoA from a fat and run it into the Krebs cycle, you can still make ATP even if you don't have glucose. Now, amination, transamination, and deamination, I'll talk about that in a second, because those don't necessarily mean using amino acids, although we can. We can use other sugar sources and run them into glycolysis just by converting them around. We can take proteins. So let's look at the basic structure of a protein. We've got the basic structure of a protein up at the top here. We've got the carboxyl group on the left, we've got the amine group on the right, and we've got 3 amino acids represented there because we've got 3 R groups. Now, if you were to break them down into individual amino acids, and you were to pop off the amine, okay, deaminate it, that becomes pyruvate. Right? We got to get rid of the R group too, but that becomes pyruvate. That is pyruvate. You can send that to the Krebs cycle. So we can actually burn protein too, and oxaloacetate. And while we're at it, when we're talking about rearranging amino acids, like I hinted at before, certainly you can deaminate it and turn it into pyruvate. You can also move the amine group around. So B here, transamination. You can start with something and turn it into an intermediate of the Krebs cycle, or you can turn it into a different amino acid. So moving the amine group around on amino acids means that we can have different products going into the Krebs cycle, or we can take different intermediates out of the Krebs cycle and use them as building blocks for amino acids. And here's that catabolism of fats. Right? So converting the fatty acid into acetyl-CoA, which would then go into the Krebs cycle. So I don't necessarily expect you to know all of those very detailed slides, like, oh, we're going to take the fatty acid and turn that into acetyl-CoA, and that's what goes into the Krebs cycle. But I want you to not forget that when we're talking about metabolism, and we're talking about the sum total, and amphibolism means using different building blocks. So different starting molecules for catabolism and catabolic products as building blocks for anabolic reactions. So when we're talking about glycolysis and pyruvate, you can actually have a lot of things. You can respire it, you can ferment it, or you can use it as an anabolic building block. And now I'm going to end this video and wrap it up with just a very short little miscellany video at the end here. So ending this one, and we'll move on to photosynthesis for the next one.